I totally forgot how draining hospital rotations are. It's my first day of internship, and it's man down. Give me a second. Hi guys, welcome to another episode of Life with Juan. It's me, who else would it be? Juan. And today we are going to be talking about arachidonic acid. Now, as you might have noted from the start of this video, prior to the intro, internship has begun. It's just my first day and everything has already been so tiring. Maybe I'm just not used to it after the break, but I figured that it would be hard to do post-production on these videos, so I won't be able to do drawing as extensively as I would want to. However, that's not going to stop me, and instead I'm going to be using PowerPoint to present my lessons and my learnings to you guys. Think of it as, those Let's Play videos where people play video games in front of you. Well, I am going to peg this Let's Study videos. Actually, these videos are just so that I can study my lessons and get more acquainted with my anatomy, physiology, biochemistry, pharmacology, all of that med stuff, right? Because I enjoy studying and I want to show you guys. I want to share my enjoyment with you guys. So anyway, let's start with the video. Arachidonic acid is a 20-carbon fatty acid. It's polyunsaturated, meaning it has 4 double bonds. You see up there? You see that picture that I made? It has 4 double bonds. They're at carbons 5, 6, 8, 9, 11, 12, and 14, 15. I still remember my biochemistry. So it has 4 double bonds. I like to think of the mnemonic arachnids have 4 pairs of legs. Likewise, arachidonic acid has 4 double bonds. And this is made via the action of MALONYL-CoA and linoleic acid. MALONYL-CoA is formed from acetyl-CoA, while linoleic acid is formed from your diet. And this is stored in your cell membrane, attached to phosphate to form a phospholipid, and it's released via phospholipase A2. That enzyme is inhibited by steroids, which is why steroids have an anti-inflammatory effect, because a lot of the downstream products of arachidonic acid are inflammatory in nature. Anyway, so arachidonic acid, the precursor of eicosanoids via either the cyclooxygenase pathway or the lipoxygenase pathway. Eicosanoids are essentially 20 carbon molecules that have roles in inflammation and coagulation. So it all starts with glucose, which you get from your diet. You get it from foods like rice, pasta. A lot of it you find in junk food, and you should regulate that if you don't want obesity, diabetes, atherosclerosis. Usually too much glucose is the culprit of that. However, in moderation, glucose is fine. Diet is always the first way to lose weight. But I digress. In your cells of your intestines, when it's absorbed via insulin, that rhymed, glucose via glycolysis is converted into pyruvate. Pyruvate is then shuttled into your mitochondria via carnitine, right? And then via either pyruvate carboxylase or pyruvate dehydrogenase, it's converted into either oxaloacetate or acetyl-CoA. Now, you remember that MALONYL-CoA is formed from acetyl-CoA. However, it has to be outside the mitochondria. So we need to find a way to get oxaloacetate and acetyl-CoA back into your cytoplasm. And how do you do that? You turn these two into citric acid, but you prevent citric acid from entering the citric acid cycle by shuttling it out of the mitochondria via the citrate pyruvate shuttle. It exchanges citrate for pyruvate, essentially. And once citrate is in your cytoplasm, we can start with forming MALONYL-CoA. In your cytoplasm, it's exposed to citrate lyase and gets a little energy from adenosine triphosphate to be converted back into acetyl-CoA and oxaloacetate. Oxaloacetate via NADH is converted to malate, which then, via NADP+, is converted to pyruvate. Pyruvate then re-enters your mitochondria. Acetyl-CoA is converted via acetyl-CoA carboxylase, some energy from ATP, and carbon dioxide from biotin into malonyl-CoA. Acetyl-CoA carboxylase is upregulated by the presence of citrate and by insulin, and it's downregulated by palmitoyl-CoA, which is a downstream product of fatty acid synthesis. The recurring theme a lot of times in biochemistry is the negative feedback. A downstream product in increased supply will inhibit actions of the upstream chemicals. And so you see it here, palmitoyl-CoA, a downstream product of malonyl-CoA, will prevent formation of malonyl-CoA. So acetyl-CoA carboxylase is active when it's dephosphorylated, and dephosphorylation happens via the enzyme phosphatase. When it's inactive, it has a phosphate group attached, which is essentially phosphorylated acetyl-CoA carboxylase, right? It's all putting stuff together. Let's talk about malonyl-CoA first. I'm too excited. Malonyl-CoA is the carboxylated form of acetyl-CoA, which comes from the citric acid cycle. And you need it in your cytoplasm to be able to use it for fatty acid elongation. It essentially adds 2 carbons to a fatty acid, and it can be used for either de novo fatty acid synthesis or synthesis of arachidonic acid. So now let's talk about linoleic acid. Linoleic acid, you get it from your diet. I'll put my image up there and tuck here. It's an 18-carbon fatty acid chain, and it consists of 2 double bonds. It's an essential fatty acid. You can't get it from de novo fatty acid synthesis, so you can't make it from malonyl-CoA. Instead, you need to absorb it from several foods like plant oils, walnuts, peanuts, cashews, and tofu. Now, once you have it, via delta-6 desaturase, it's converted into gamma-linoleic acid. This, combined with malonyl-CoA, elongates to form dihomogamma-linoleic acid. This, via delta-5 desaturase, the enzyme, some oxygen, NADH, and hydrogen, is turned into our 20-carbon friend, arachidonic acid. Now, arachidonic acid, via cyclooxygenase, the enzyme, and 2 oxygen molecules, is turned into prostaglandin G2, which then, by peroxidase, and 2 glutathione molecules, is turned into prostaglandin H2. The downstream products are this of prostaglandin H2 are all prostaglandins or thromboxane. Anyway, the cyclooxygenase pathway is catalyzed, as I said, by cyclooxygenase, which is involved in cyclization of arachidonic acid prostaglandins. It puts a little ring. It puts a ring on it because it likes it. You get the reference. You see what I did there. Anyway, irreversible inhibition of COX is done by aspirin. That's why you give, one of the downstream products of cyclooxygenase pathway is thromboxane. And thromboxane helps in coagulation by causing aggregation of platelets and constriction of your vessels. Sometimes this is bad because it can block blood flow to your brain, and that's why you give patients an irreversible cyclooxygenase inhibitor, namely aspirin, so that you can restore blood flow to your brain. Likewise, it's reversibly inhibited by acetaminophen and NSAIDs, as well as celecoxib. That's why you give NSAIDs, acetaminophen, and celecoxib sometimes for fever and for pain. Then, peroxidase is the enzyme that cleaves an oxygen-to-oxygen bond to form prostaglandin H2 from prostaglandin G2. Now, thromboxanes, our first product, is formed via thromboxane synthase, which you find a lot in your platelets. Now, as I said in our last video, thromboxane is important for vasoconstriction, platelet aggregation, as well as proliferation of lymphocytes and bronchoconstriction. Those last 2 I didn't actually mention since I was focusing on platelets. Then, we have prostaglandins I2, E2, and D2, which inhibit inflammation. These namely inhibit IL-1 and IL-2. These inhibit platelet aggregation. These inhibit leukocyte aggregation. These inhibit migration of lymphocytes and proliferation of T cells, while these promote vasodilation and cyclic AMP. Cyclic AMP essentially helps your blood vessels to dilate. Then we have PGF2 alpha. There should be an alpha there, which promotes vasoconstriction, bronchoconstriction, and smooth muscle contraction. So this one promotes vasoconstriction, unlike the prior prostaglandins. Okay, you guys good? You guys good? Let's go to the next set of pathways, the lipoxygenase pathway, our last topic for today. Lipoxygenase converts arachidonic acid into HPETE, and it's inhibited by xylutone. So xylutone is a drug that you give in order to inhibit inflammation. And because of this, HPETE can form either lipoxins or leukotrienes. Let's talk first about lipoxins. These inhibit edema, angiogenesis, and prevent reperfusion injury. Okay, let's talk about these. What's edema? Edema is when water leaks out too much of your blood vessels into the third space, causing you to balloon up with water, like a walking water balloon. And so these patients, their feet are really huge, and that's what you call edema, essentially. Then it also inhibits angiogenesis, namely the generation of new blood vessels, and these prevent reperfusion injury. Reperfusion injury is when an organ has been starved of circulation for a while, and then it has a sudden rush of blood. And with that sudden rush of blood, it gets damaged, it gets inflamed, it gets oxidized. That's reperfusion injury. Now, leukotrienes. These are LTC4, LTD4, and LTE4. These all are essential for inflammation, smooth muscle contraction, area constriction, and permeability of your capillaries, which allows your white blood cells to move through for chemotaxis. And you give, hence, leukotriene inhibitors to patients who have inflammatory reactions, namely asthmatic patients. So these are inhibited by montelukast and zafirlukast. You give leukotriene inhibitors to patients with asthma to prevent the bronchoconstriction and to allow them to breathe, essentially. Then finally, HETE and leukotriene B4. These promote chemotaxis, adenylyl cyclase, and degranulation of lymphocytes and leukocytes. So LTB4 specifically promotes neutrophil adhesion and promotes suppressor CD8 and inhibits CD4 lymphocytes. So CD8 suppressor is essentially a regulator of inflammation. It's like, hey, wait up, we're not ready to go full on war yet. And CD4 lymphocytes essentially are in charge of releasing, of recognizing enemy cells as presented by antigen-presenting cells like macrophages and dendritic cells. HETE is also formed from the cytochrome pathway, which is cytochrome 450. Okay, that's the last part of this video. Essentially, we talked about arachidonic acid, how it's formed. We talked a little bit about formation of malonyl-CoA. We talked about linoleic acid and getting it into your body from your diet. We talked about cyclooxygenase pathway and the lipoxygenase pathway. And that was my video. References are the textbook of Biochemistry by Devlin, TM, First Aid for the USMLE Step 1 by Le, and Mark's Essentials of Medical Biochemistry by Lieberman and Peet. Thanks so much for watching this video. I hope you all enjoyed. I hope you all learned a little something. I hope that I'll be able to continue doing it this way. It helps me study and it helps me be productive, and I hope it helps you guys as well. If you like this video, please leave a thumbs up on the video. Please subscribe, hit the bell, stay in tune for future videos, and leave a comment in the comments below. Remember, live life to the fullest because you only have one life to live.
Word of the day.
Arachidonic acid.
Na 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 na.